iGEM is an opportunity for students who get the chance to construct mind-blowing and half-made experiments using modern techniques in biotechnology. iGEM is a worldwide competition in creating the most exciting and innovative living constructs by combining genes from different life forms. The purpose of the competition is training students in combining basic research and practical applications to find creative solutions to actual problems. In this year's iGEM competition, 193 teams from around the globe are competing to make the most innovative project. Teams must also consider the ethical issues arising from the creation of new organisms. Students who participate in this competition spend their summer holiday in the laboratory working on their project. Students have an opportunity to work on a self-chosen topic, and the university provides all necessary facilities and equipment.
I am a member of a 10-man team. My function here is I'm in charge of laboratory protocols.
What is a laboratory protocol?
Well, when we do these experiments, we need a plan, and I'm in charge of making sure this plan will work. It involves looking up theoretical stuff, reading articles, constructing ways to do these experiments from already acquired knowledge shared on the internet and databases. This whole project is a major learning experience. It's a great opportunity to learn techniques and gain some lab experience, some real lab experiments with a project we decided on ourselves, with the protocols we developed ourselves. It's just a really great learning opportunity.
And how old are you?
I'm 26.
Okay. So you think you're the oldest in the group?
Yeah, yes, I'm the oldest living member of this group.
My function in this iGEM team is to, well, basically it have been idea development. So I was the one who proposed the idea in the beginning, and the others thought it might be a good idea, so we looked into it. And then every guy came up with different aspects of the idea, and we slowly developed it. So I have mainly been in charge of the background knowledge and the theoretical stuff that needed to be taken care of in order to understand the idea and the project we're going to do. So I am 20 years old, which makes me the youngest member in the group.
I am doing a lot of different things. I am making travel arrangements for Bielefeld and Uppsala. We are going to a conference there, and in a project like this we have to not only be in the lab doing science, but there's a lot of other things to do, like marketing, and I'm contacting the press, the media. From this project I expect to get a lot of experience being in the lab, and that I can use in my next job when I get out of the university.
To watch the students participate in iGEM is really to watch a transformation where the students go from being students to fully grown academicals, because they're pulled through all the different stages. They have to get their own funding, they work with their own project in the lab, they have to work in teams. Often it's teams where you have people from different areas of study, and it really challenges them.
Internationally Genetically Engineered Machine, iGEM, is a contest in synthetic biology where organisms, like bacteria and yeast, are perceived as microscopic machines and are programmed to produce simple or complex molecules. This programming is done through the controlled insertion of DNA into the organism of choice.
What we just did was to spin down all the larger parts of the cell, which you can see in the bottom is a very little pellet, we call it, which is the DNA and the proteins and every large compartment of the cell. So now we can remove all the liquid and start to extract the DNA.
DNA can be transferred from one organism to another. In this way, a third new organism is created.
That will have some very beneficial effects on your digestive systems. You will feel more satisfied by eating less. You won't gain weight. It might reduce fat mass development and might reduce the risk of colon cancer.
So now the liquid is all up here, and we want to spin it down through the filter which is placed in the middle. And this filter is positively charged, which means it can bind the DNA. So when we spin it, only everything else will go down through this tube, but the DNA will stay in the membrane. And we will keep on washing stuff through except the DNA, so we can separate the DNA from everything else. And when that is done, at last we will take a liquid, an elution buffer, that can take the DNA with it through the column. So we will end up with only elution buffer and DNA in the bottom here. This is the last spin, yeah. So now we should have the DNA together with the elution buffer that we just added.
I'm working here as a professor in molecular oncology, and besides that I'm also leading a large center of excellence. That's a center of excellence in nanomedicine research, where we aim at developing new medical devices for treatment of cancer based on nanotechnology principles. I became an iGEM advisor because I found the idea of the competition quite interesting. And I found it worthwhile to work together with the students on a project they have been, so to say, inventing and which they are organizing themselves.
It costs the university quite a lot of money to participate in iGEM, but what they get are these really mature students who we've seen in the previous years really glow. They find really exciting projects out in the world or here at SDU, and they often excel as scientists later on.
We just mixed a mix of DNA and our restriction enzymes. And we want to take that mix and put it in this gel I'm about to make. And then we can separate the DNA by length. And we have a specific length of a fragment that we want to get. And in order to find that fragment in the mix of all the DNA that's being cut, we need to make a gel. Because then it will separate the DNA fragments when we run it. And the reason that we add this ethidium bromide is because it binds to the DNA and glows when it's lighted on with UV light. Now it fits. Yeah. Alright, so we put a comb in here, which you can see when we put this in before this solidifies. We will pour it into this chamber. And now, in about 10 minutes, it should be solid or gooey. So now you see it's solid. And we will put it in this chamber. And now I just need to get our DNA ladder, so we will know how far the fragments have gone in the gel. Alright, so now we have transferred the DNA and this gene ruler, which consists of different sizes of fragments that allows us to see how long our DNA is. So as you can see now, I have started the electricity and we'll run it at 100 volts, which is what fits for this size of gel. And now we'll just wait, and the negatively charged DNA that is loaded in one end of the gel will wander against the positive pole. So it will wander through the through the gel, and the smaller the fragment is, the faster it will run. So by time we will separate the sizes of the fragments, and hopefully we will find our gene. This circle indicates the DNA that we have cut two places with two enzymes. That is what we just did. So we cut it here and here. And that leaves us with two fragments, the one that's here and the one that's here. And those two fragments is what we see here on our picture. Okay, so the larger fragment would be the 2/3 of the circle, approximately, and then the last smaller fragment would be the little circle here that we cut out. So that is what we just did, and we want to isolate the gene, which is the small fragment. So now we're going to cut that out of the gel. This UV bench will shoot UV light up through the gel now, and because we added the ethidium bromide, it will glow where there are DNA in this gel, so we will be able to see what we need to cut out in order to isolate our gene from the rest. And that I will do with this regular scalpel. And let's turn off all lights. Here we are. Okay, so now you see, as the picture before, there's two glowing dots, which is the rest of the plasmid, the larger one here furthest away, and the closest is our gene. So I'm going to cut out our gene simply by cutting down in the gel like this. Okay, and now I'll turn off because the UV hurts the DNA and we don't want it to mutate more than absolutely necessary. Okay, so now I can take up this piece of the gel which will contain our gene, and I just need to get it in this tube. Here we go. So the final product, a little piece of gel, hopefully with our DNA in it.